Hey, I'm Dr. Rob. Welcome to Biblical Genetics. I am on my back porch today enjoying a beautiful, cool May morning. I have a day off because I'm going to Tennessee this weekend, so it's a Friday. I've got the whole day to myself. I said I need to film something. In fact, this is a second filming. I tried to do this episode 2 weekends ago. I was on the border of Virginia and West Virginia, speaking of some churches up there, and I was staying at a friend's house, and they're literally right next to the Appalachian Trail. So on my way out Monday morning, I pulled off at one of the trail access points, and I walked up to something called the Kefir Oak. It was a local landmark, and all the locals were like, oh, the Kefir Oak, the oak tree, oh, blah, 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 blah. And they're all like super impressed. And so I got up there, and I was going to film my episode, and I wasn't very impressed with the tree. It wasn't as big as I thought it should be for an impressive tree. I've seen bigger oaks. It's the second largest oak tree on the Appalachian Trail. Okay, it doesn't mean it's the second largest oak tree I've ever seen. In fact, I've cut down an oak tree larger than that. It was dead. It was on one of my neighbor's properties, and he asked me if I would like to take this tree down. I said, yeah, sure. I needed some firewood. I didn't realize which tree he was asking me. This thing was 6 feet wide at the base. It was like 90 feet tall. In fact, I've got some examples of boards left over from this tree. This is about 15 years ago. I had several years worth of firewood. I made a floor out of it. I made molding out of it. I made a table out of it. I made some of the shelving that you've seen in some other Biblical Genetics episodes out of it. It's just woodworking is a hobby. It's a fun hobby. In fact, today I've got a laser engraver that someone lent to me going, and I'm doing some experiments. I made a little kitty cat. That was my first one. That's pretty good. And I tried to etch a picture of my daughter on this board, but it did not work. It's just black. So my beautiful daughter did not come through. I'm going to try again. But this is fun. I love working with wood. It's interesting, sort of. Actually, it's not. Wood's actually kind of boring. It's beautiful, sure. But it is just a little bit boring because, what does a tree do? It exchanges carbon dioxide with the atmosphere. It releases oxygen. It releases water. We figured out how trees work a long time ago. But it takes that carbon dioxide and makes wood, and that's fascinating enough. I mean, the production of wood is really cool, chemically, kinda. I know people get their PhDs in, wood, but I'm glad I didn't get my PhD in wood. I think I would have been bored to tears. The one thing that trees do, though, which is absolutely amazing, is photosynthesis. Photosynthesis is simply the least predictable chemical reaction to happen in the biological world. It should never happen. It's too complicated. You're talking about biological molecules that are capturing photons that should destroy the biological molecule, but they're all lined up together so they can do a hot potato, hand off the energy from that photon to a series of chemicals so that any single chemical doesn't get destroyed by the energy. And they pump hydrogen atoms across a membrane. They use that to make ATP, and the ATP is used for all the energy of wood production. Okay, that's fascinating. But, I don't know, are trees really that interesting? Above ground? See, this is my bridge point where I want to talk about what happens in the cell in biology. Most of the interesting things that happen in a forest happen underground. And these are things that remained hidden from us for a very long time. It's since the dawn of humanity we've known trees. It didn't take too long to figure out that trees grow tall, they grow wide, they add mass over time. Then we figured out that they absorb carbon dioxide a very long time ago and make that into wood. But in recent years, in recent decades, we've been learning about this incredible ecosystem under the ground. I mean, it's like a civilization under the ground. The trees are talking to other trees of the same and different species. They're talking to plants. They're talking to fungi. They're talking to bacteria. And they're like, hey, Mr. Fungi, you have any molybdenum? I need some molybdenum. I got some sugar. And they're doing exchange of minerals across the tree root as the tree is communicating with the things in the soil. In fact, the things wouldn't even be in the soil if it wasn't for the presence of the tree. So all this really cool, amazing things have remained hidden for us for a long time. And that's my bridge point to talk about the world of RNA, which has remained hidden for us for a long time. Now, I got slammed on one of my recent videos. Now, granted, I don't pay attention to the comments on most of my videos because, I know there's just a bunch of trolls out there, and it's not worth my time to engage with people who aren't going to fairly engage back. So I don't usually pay attention to the comments. But one particular person was ragging on me for basically rejecting neo-Darwinism, the thought that you could have a mutation in a protein coding gene that creates a new trait or a new phenotype that natural selection can then act upon. And I'm like, dude, man, that's so 20th century. I mean, this is the year 2023. You're talking about 1920s, 30s, 40s, 50s, 60s ideas. Now, granted, back in the day, we didn't know how things worked, and life seemed kind of simple, and it kind of made sense that if you had a mutation in a protein coding gene, that would do something that natural selection could act upon. Okay, fine. But they realized pretty quickly that most mutations don't have a giant effect. And then they realized in the late 60s that only about 2% of the genome codes for protein. So in the early 70s, they came up with the junk DNA hypothesis that, oh yeah, well, most of the genome can mutate at random, but only the important bits, the protein coding regions. Mutations happen there. That's where selection can either amplify or reduce a trait based on if it's good or bad for the organism. Yeah, that didn't work too well in the modern era because now, after like the ENCODE project and multiple other things, we realize that most of the information in the cell is in the non-coding regions, the so-called junk DNA regions. And so, I can't fault someone for not knowing what I'm about to share with you because, I'd learned a lot of this myself, like this month. I've just spent some time doing a deep dive when I'm not working with wood or other projects, studying RNA. I read multiple scientific papers, multiple web articles, and one incredibly important modern book, which I'm going to be reviewing for the Journal of Creation when I'm finished with it, which is extremely laborious and hard to get through because it has so much detail. And learning things, shocking things, learning more things, learning things that are just overwhelming. There's so many different types of RNA that it's really hard to keep up with it, but it is abundantly clear that the neo-Darwinian thought that you can have a simple change of a letter in the genome that natural selection is going to act upon is moribund. It's passé. It's an old idea that doesn't really work anymore because there's so much more information, and the information is at different levels. I remember Dr. John Sanford's book, Genetic Entropy and the Mystery of the Genome. He had this brilliant illustration, the Princess and the Pea illustration, where he said the mutation is like a pea underneath a mattress, underneath another mattress, underneath another mattress, underneath another mattress, and the organism, or the princess, is sleeping on top of the mattress. And if she can't detect that mutation, then natural selection can't do anything. And it was a brilliant idea because you're talking about environmental differences, other genes that are affecting whether or not this mutation has any effect, epigenetic differences, exercise, diet, even attitude and spiritual, psychological sort of things all come into play here when you're talking about whether or not a single mutation can produce something that natural selection act upon. So the princess and the pea and all the stacked up mattresses is brilliant. But that was an idea that can be even put on steroids when we talk about RNA. Most of the work done in the cell is on the level of RNA. Most of the information is packed into the non-coding regions. Most of the control of the genes happens with RNA. For example, as I'm in graduate school, I'm learning about transcription factors. These are proteins that attach to DNA just upstream of a gene. And if they're there, they can basically trigger transcription of the gene, which then can be turned into protein. But transcription factors have a very small recognition window, and they're not very specific. They can theoretically stick at other places in the genome. So how come they only stick there? Well, now we learn that there are RNAs called enhancers that will attach very specifically to the DNA and then grab the transcription factor and stick it onto the DNA so that the gene can be turned on. So in the end, it wasn't the proteins that were doing the work. It was the RNA that was doing the work. There are many, many other examples like this. You probably are familiar with messenger RNA. We'll start with that one. When a gene is made into RNA, it's made into something called messenger RNA. Now, in higher organisms, our introns have to be cut out, and then the exons have to be joined together, and then that can be translated into protein in the ribosome. But the messenger RNA is basically what's going to turn into a protein. And then the cell uses things called transfer RNAs. A transfer RNA has a little 3-letter code at the bottom called a codon, which sticks onto the messenger RNA in a specific 3-letter spot. And on top of the transfer RNA, there's an amino acid. The amino acid is added to the growing protein strand, and that's how we get a protein. But the machine that does that, the ribosome, is a very complex combination of proteins and RNA. So we have ribosomal RNA, we have transfer RNA, and we have messenger RNA. Those are RNAs most people are familiar with. But now we know there are things called microRNAs. There are long non-coding RNAs, or link, LNC RNAs. These are the most important thing. I want to talk about these specifically. Because when they were first discovered, nobody knew what to do with them. They weren't supposed to be there. The information in cells is supposed to be in the protein coding regions, not in this other stuff. So they just said, oh, these are long non-coding RNAs, and they threw them into a basket. And now we're learning that there's lots of different types. In fact, they should not all be called the same thing. They should forget that long non-coding designation, throw it away, and let's talk about specific types of RNA. Because there are some long non-coding RNAs that are a couple hundred letters long, and there's some that are tens of thousands of letters long. There are entire long non-coding RNAs that look and act just like protein coding genes. They have introns and exons. They're alternately spliced depending upon the environmental conditions or the state of development of the organism. I mean, they look like genes, but you can't take the information there and turn it into a protein. It would be a nonsense protein. It has nothing to do at all with the production of proteins, and yet it looks just like a gene? What is that thing? One of them in particular is called Exist. It's like 17,000 letters long, I think, and it's got, 8 exons. Incredibly important thing that's happening in the cell, and we knew nothing about it. There's a couple of reasons for that. The early geneticists can't be faulted for focusing on DNA. It was easy to sequence. It's a much more stable chemical, and it's just easier to work with. One of the problems with DNA, or RNA, I should say, is that it has a lot more than 4 letters. Now, DNA, A, C, G, and T. And sometimes the C's can be methylated, so you have another letter there also. But it's really just A, C, G, and T. Adenine, cytosine, guanine, and thymine. Well, RNA has A, C, G, and U. Instead of thymine, there's a uracil. But that's only when it's first transcribed. Hundreds of chemical modifications happen to RNA. One of the big things we see in RNA is inosine. Take an adenine and translate it or chemically modify it into inosine. Inosine is really common in RNA, but so are lots of other modifications. And every time you modify one of the nucleotides, it's a different nucleotide. It's a different chemical. And so sequencing RNA is really hard. One of the reasons why RNA technology was held back is, besides the fact that it's an unstable molecule, it's not as easy to extract. It's really hard to sequence. Well, recently people have turned the nanopore sequencers on RNA. Nanopore sequencing is amazing. You take a charged plate with a little micro hole in it, and you can grab a piece of DNA and pull it through the hole. And on this charged plate, as the letters are going through, the charges on the A, C's, G's, and T's, you can read them and say, say A, C, 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 G, 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 T, C, C, A, C, C, G, G, T. Oh, that's really amazing. Well, they've been tuning that to work better with RNA now. And so they haven't figured out all of it, but a lot of the letters that we see in RNA can now be sequenced directly. And that has been a huge boon for RNA technology, because now we can see what's happening in the cell a lot more easily. But it was slowed down. I mean, I remember doing, extractions of nucleotides from corals and bacteria, and you could see RNA in there. You just ignore it. It would just be in a different spot, be like blobbed up on the bottom or the top of your gel. Just forget that. That's the RNA area. Stay away from that, because no one knew what to do with it. But now we have a much better idea that is very important. So we have long non-coding RNAs. These things, they help transcription factors. They help control alternate splicing of the genes. They are heavily edited, especially the A to I. They're also methylated. There's one called ADAR1, which binds near repetitive elements and helps fold up the DNA. All those repeats that are in the junk DNA, those things, the ALUs and the things that are just randomly scattered in the genome, it turns out that they're very important. That a lot of non-coding RNAs will match up to a lot of these repeat sequences and cause folding and bending of the DNA. Another thing RNA does, it It can form triplex structures with DNA. It can lay down in the major groove of the DNA double helix and interfere with transcription. So it can turn genes on, it can turn genes off. Non-coding RNAs can also cluster in the cell to make what are called phase-separated domains. Just like if you mix oil and water together, it'll separate by phase. Well, the same thing happens if you have a bunch of RNAs that are similar, clumped together. They can literally cut off the outside of the cell and make a little pocket all by themselves, almost like an organelle, but it doesn't have a membrane around it. And the inside of that pocket will have a different chemistry than the cell itself. So all these little micro spots inside the cell that are doing all these very strange chemical and biological genetic things because of RNAs, which turn out to be extremely important again. Non-coding RNAs affect cell fate. They control memory. They modify histones, the proteins that DNA wraps around. Well, histones have a long tail that can be heavily modified. They're often modified sometimes 50, 20 times. RNAs tend to do that. There are modification writers, modification readers, and modification erasers amongst the non-coding RNAs. They do incredible things in the cell, only just learning this now. We also now know that RNA can be inherited from one generation to the next. That is modified RNA. RNAs that are modified by environmental or cell state conditions can be passed on. Now, chromatin is DNA that's wrapped around histone protein, like beads on a string. We've known this for years, but what we now understand is that chromatin is absolutely coated with RNAs. RNAs that are passed from one generation to the next. In zebrafish, the methylation state of the sperm is preserved. The methylation state of the egg is erased. And then after fertilization, the female contributed DNA is methylated according to the male contributed DNA. So whatever genes are turned on and turned off in the male are turned on or off in the babies that grow after fertilization. That means that information is passed from one generation to the next that's not in the DNA. That means that there's not a absolute difference between the reproductive cells and the body cells. That's called or the Weissman barrier that was thought of in the 1800s. Well, that's not true. In fact, now we're talking about Lamarckian type inheritance, where you can have the inheritance of acquired characteristics. That's not supposed to be true. That's not Darwinian. And that just makes the situation much, much, much more complicated. And that's the cool thing about biology, how complicated it is. Because I've said this a couple times. Let me say it one more time. If life were really simple, evolution might be possible. But the more complex life is, the less possible it is for it to evolve. The more likely it is that it was in fact designed by an incredibly intelligent designer. Now, that's not a proof. That's not, there's no proof in science anyway. But it's a statistical argument. The more complicated things are, the more likely it is that it had to be engineered. And so the way I imagine this is when God's inventing his life forms, he takes a cell, he puts all the proper components in there, he winds it up, he adds a little bit of energy, gets everything in the proper area, proper location, then he lets go. And then it's alive. Without that sort of an input, an intelligent input from a higher power, you're not going to get life. And if you did ever get life, which is not going to happen, but if you ever did, it would remain simple. Life is not going to figure out improbable chemical reactions. It's not going to figure out complicated ways to regulate information. It's going to remain simple, remain fast reproducing, and small with a compact genome, because the things that have parasitic elements in their DNA are going to reproduce more slowly. They should be selected away. One of the things that they thought of with RNA when they first realized that cells producing a lot of RNA that doesn't code for proteins, well, they called it parasitic RNA. Oh, that's just the junk. See, that's just the stuff that infected us millions of years ago that we're still producing, and that's not important. Oh, wait a minute. In biology, form follows function is a very important idea. Many, many times, if someone finds something, it's okay, there's something there. What does it do? It must do something. In fact, a lot of the things they thought were vestigial in the cell turn out to be not vestigial. And I think that junk DNA, that whole argument, is the vestigial organ. It's a modern vestigial organ argument. Back in the 1800s, they had a list of 80 different things in the human body that don't do anything. Well, that's not true. We found functions for all of them. And now we're finding more and more functions for the so-called junk DNA. because it's not junk. It never was junk. In fact, it's always been important, but evolutionary bias made people think that it's actually just nothing. Now, that doesn't mean that every single thing in the cell has a function. That's not true. I mean, it doesn't have to be true. We could have parasitic DNA. We could have broken DNA. We could have DNA that's just there for scaffolding. It doesn't have to actually have a specific function biochemically or genetically, but most of it should be functional. In fact, in evolutionist, you probably should think that most of it's functional, because selection should always be driving to get rid of the parasitic stuff, the broken stuff. Optimization should be happening over billions of years. And yet, at the same time, you can only optimize what already exists. You're not going to think of, evolution is not going to think of strange, new, bizarre things that come out of nowhere that would never be predicted based on simple chemistry of the progenitor. You're not going to get complex chemistry, complex genetic, complex structures that are tightly regulated in complex ways through a simple trial and error process. So honestly, I think that the design idea, the thought that there's a God that created life, is a much more powerful explanation than the evolutionary model. Now, granted, evolution does explain a lot. It sure does. There's a lot of things in evolution that seem to fit very well with the living world. But that doesn't mean that it is the answer. And when you look at what evolution cannot explain, that's where you see the failure of evolution, not with what it can explain. That's not how science works. In the same way, in creationism, I know that me and my colleagues, we spend a lot of time trying to find things that match up with what our beliefs are. Fine, we do that. All people do that. I'm just a fact of human nature. But I'm also trying to find areas that I cannot explain. And there are some I can't explain. Sure. Yep, look at that right there. That sure looks like an evolutionary thing. But what I've learned from experience is that when you take a deep dive and look at that thing, like junk DNA, parasitic RNA, non-coding RNA, you'll very often find that the story that you're first told isn't the real story, and that it's much more complicated, much less likely to have evolved than anyone at first thought. So there you have my second attempt at explaining some of the hidden mysteries of the cell and things that are arguing against a very simplistic belief in Darwinian processes. The cell is much more complex than really even me, than anyone imagined, and therefore it was designed by the God of the Bible. There's more coming. I'm going to film another episode on my way up to Tennessee this weekend. I'm going to be talking about why you can't trace your family tree back to Adam and Eve. Yes, we do come from Adam and Eve, but you can't actually prove it genetically or genealogically. And that'll be a fun one. We'll see how well I do. I might have to come back on my back porch and film it again. We'll see what happens. Trying to make short movies on the road is really hard. There's all sorts of things that can go wrong. The weather can go wrong. I'm very often forgetting things, or something's not charged up, or something breaks. But I love doing this. I love talking about God's creation. I love encouraging people, and I love explaining things to people that maybe we didn't quite understand as we're going through school. Especially love contradicting things that were taught in textbooks. It's funny because textbooks are already 10 years old before they come out. And then if you've been out of college for like 10 or 20 years, I mean, you have 20, 30-year-old information in your head. And the world has changed. It's complicated. It's designed. It's designed by God. Hey, thank you so much for your support. I really appreciate the people on Patreon and the people on buymeacoffee.com that are contributing to help encourage me to keep doing this. My costs are basically covered, and that's a great thing. If you want to help contribute, there'll be links in the show notes. So I'm not asking, I'm just saying that it's there. Oh, by the way, you really want to help, really want to help, click like and share. That's how the internet works, and I'd appreciate it very much. You'll have a wonderful day. Good cow. Wow, I hope they like me. That's a big old, hey, beast. Good, they're walking that way. I hope they don't turn around, because my second camera's over there, and I have to run and grab it. Yeah.